Oh, wow. Welcome, everyone. Glad you're here. This is coming to a close. This is the very last section. I did do the work problems on Teams on Thursday, so you can get the work problems explained. If that helps you learn better, all the more power to you. It's good to have that human interaction. Um, this is the last recording that I'm going to do, I think, and the course has been great. You students have been hardworking and driven, and that's all good. And so we're going to talk more about enzymes and things that factor into how enzymes are affected. You already know some, you know that heat and acid and agitation and all that affect enzymes, but we haven't really thought about substrates, or we haven't thought about inhibitors or things that biologically regulate enzymes. We thought about external factors like heat and acid and base, but now we're going to think about how the body may get into regulating these enzymes. So they slow enzymes down, or they make enzymes work faster. They block enzymes, drugs that can block enzymes, all these things, very important. Penicillin, that's all about blocking enzymes in bacteria and such. And so let's go ahead and move forward. Okay, activity of enzymes. It describes basically the speed of enzyme catalysis for reactions that convert substrate to products. And if you take more chemistry, you'll learn where you'll actually get a turnover rate and a K-cat and all these really cool things we're not going to get into. It's not going to help you at this stage. But um, reaction conditions, as we learn, temperature, pH, are going to affect an enzyme. But we haven't talked about inhibitors. And guess what? There are inhibitors that can sometimes block the active site, or sometimes they don't block the active site where the substrate binds. They actually attach to another part of the enzyme and cause the enzyme to act crazy and change its shape, and therefore the active site sort of disappears or becomes misshaped and cannot bind its substrate. But a kind of a just a quick deviation, because I like diversity and I like learning about animals and plants and the earth and everything in science, and I'm sure some of you do as well. This is kind of a deviation, but we know that heat can mess up enzymes. We know heat can destroy enzymes and the tertiary interactions between the R groups and even further. But we do have some exceptional um, things that can survive in really high temperatures, which means there's some biological chemistry going on, kind of like a superhero that can walk through fire, right? Well, thermophiles are that superhero because they can survive in high temperatures, like 50 degrees Celsius to 120 degrees Celsius. And we're at 37 degrees Celsius. We wouldn't do so well in hot springs, right? We don't want to dive into a hot spring, right? But there are bacteria and organisms that can survive under high temperatures. So that's the diversity of life working on our planet. And enzymes, they do have an optimal temperature. And we could make this simple graph, although I say simple, I should not say easy. Or if something is easy, I shouldn't say that it's not hard. Something can be easy and take a lot of work. Well, science takes graphs. You need to know Excel. You need to know how to work statistics. Even for nursing, you'll need some decent mathematical abilities. And some of you have proven you're pretty good at math, and I'm impressed. But we can graph this with the y versus the x. We often take the y component and we say reaction rate versus temperature. We like to always put the y component usually before the x component. So reaction rates versus temperature or degrees Celsius, right? So, um, and as the reaction rate goes up for that enzyme, we see a peak, and we see that at about 37, 38 degrees Celsius is the maximum enzyme activity, meaning it creates the most product. But if we keep raising the temperature and we get it up past 50 degrees, suddenly that enzyme doesn't want to work very well, or it doesn't work at all. So we have parameters for what the temperature needs to be for this enzyme to operate fairly optimal, right? So at 50 degrees Celsius, we're getting denaturation. Okay, so we have to be careful. As humans, have to be careful not to be out in 120 degree weather. That's not safe for us, right? And so, wow. Optimum pH values. So we're going to look at enzymes. What do those enzymes react with? What are their substrate? And where are they located? And what is the optimal pH? Because enzymes each can have different optimal pHs where they function better in an acidic environment and don't function at all in a base environment. Or they function well in a base environment and don't function at all in an acidic environment. Think about that. It's unique. It's properties of the enzyme. So enzymes in the body, many of them could have an optimum pH of 7.4, but not always. In certain organs, they will operate at lower or higher optimal pH values. So yeah, a lot of enzymes, and we're not showing all those, operate best around 7.4. But many of them are sequestered, meaning kept apart, in areas that could be very acidic or very basic. The extreme is pepsin, found in the stomach. It breaks peptide bonds, and its optimal pH is 1.5 to 2.0. Well, that's not going to be operating in the bloodstream, or we wouldn't be alive, right? Or we can look at arginase, which operates an arginine, the amino acid, and does things to that, breaks that apart. That's found in the liver, and that's very basic at 9.7. That's not going to be in our bloodstream either. So these things are compartmentalized. They are segregated or sequestered. All three of those words mean the same: sequestered, compartmentalized, um, you know, um, kind of the same meaning. Pepsin found in the stomach breaks peptide bonds. Optimal pH is very low, very acidic. Lactase found in the GI tract. It operates on lactose. Its optimum pH is 6.0. So it's a little bit acidic, but not like with pepsin. Sucrase, it's in the small intestines. It operates on sucrose, and its pH is a little bit higher at 6.2. It's still somewhat acidic. We have amylase. It's in the pancreas. It operates on the amylose sugar, breaking that down, and its optimal pH is 6.7 to 7.0. Urease in the liver, it operates on urea and breaks urea down, and it is at about 7.0. Trypsin, it's found in the small intestines. Like pepsin, it breaks down peptide bonds. It has a pH of 7.7 to 8. Different, look at pepsin, which also operates on peptide bonds. It's the very opposite. The pH is very acidic. Trypsin, the pH is sort of basic. Now we have lipase that can break up lipids like fatty acids and glycerol. That's in the pancreas. It breaks lipid and ester or lipid ester bonds, and it's a little bit basic at 8.0. And then we have arginase in the liver, as we mentioned earlier, which operates on the amino acid arginine, and it has an optimal pH of 9.7. So they work the best and the fastest when they are in these organs. These enzymes are found in these organs, and their optimal pH is set here. Now, great stuff. So enzymes and pH are very important. If we're to ever take these enzymes out of the human body, out of an in vivo system into an in vitro system, we have to have a buffer that controls for the optimal pH so the enzyme can function if we want it to function. So now we can plot reaction rate versus pH at certain reaction rates, which is um, the pH determines the rate. The higher the pH, some enzymes will not operate, and if the enzyme pH is too low, it will not operate. It has an optimal point of around a little bit over 7 in this case, whatever enzyme this is. And you could look at that table and figure out which enzymes this might be based on this reaction profile. So most active, they make the most product from the substrate when it's at the maximum enzyme activity when it's at the optimal pH. The R groups of the amino acid must have the proper charge and be in the optimal pH to perform properly, because in the active site, the substrate will bind, and the pH can affect those R groups and the active site if easily accessed to the media. So we lose activity in low or high pH as tertiary structures can be disrupted. So we don't want to lose our R group interactions. Our R group interactions really help hold the substrate in place or do the chemistry we need to catalyze the reaction. And R groups are sensitive oftentimes to pH, especially polar and acidic and basic ionized amino acids. Okay, what's this? Sucrase has an optimal temperature of 37 and an optimal pH of 6.2. Determine the impact of each change on the enzyme's activity. Will it increase or decrease the activity, or will there be no change? So we know the pH is 6.2, but say we make the environment more acidic and we change the pH all the way down to 4. That's like a hundredfold change in the acidic concentration, so it's more acid. What do you think is going to happen? Well, it's 6.2. You lower that pH, it's probably going to decrease the activity. That doesn't sound so good. So that won't help make it run faster. If we wanted to slow it down, okay, or stop it, running the reaction at 70 degrees Celsius. Well, the optimal temperature is at 37 degrees Celsius, which is like our body temperature roughly. So at 70 degrees Celsius, oh no, that's like almost twice the temperature. That's probably going to decrease the activity too, because the R groups are going to be agitated more because all the heat energy added, and that's going to be disruptive. So that's both A and B should be decreased, and that's what it is. So enzymes, remember, they're like fine. They need to be refined, and they need to be in an optimal environment, just like the student. The student needs to be in an optimal environment too, right? Don't have a bunch of worries. You can study. You don't have to work too many hours. You can relax and learn. You can push yourself, right? Not different from what understanding what optimum means. We know from our personal experiences. So inhibitors, what are inhibitors? Inhibitors do exactly what we would expect them to do. They stop the enzyme from working, meaning they stop the enzyme from producing product. So inhibitors are molecules that cause the loss of catalytic activity. They cause the active site not to function as well. They prevent substrates sometimes from fitting into the active sites, or they almost always do. There are different kind of inhibitors. There are inhibitors that actually can bind and never come off the enzyme, which destroys the protein, which you want to do to bacteria, like penicillin. You form a covalent bond at the active site from the penicillin-like molecule, and that enzyme in the bacteria can no longer function, and the bacteria is going to die because of that. Thank goodness, so that we can live, right? So enzyme plus the substrate comes together. We form the enzyme-substrate complex. It reacts and forms the enzyme, freed again almost exactly as it was when it first started, but now a product comes off. But if we have an enzyme and an inhibitor, the inhibitor can bind the enzyme, and guess what? No product, maybe no substrate can bind either. So there's lots of different things that can happen. Looking at this, let's kind of string it out and make it a little bit longer. Um, a competitive inhibitor, okay, there's different types of inhibitors. Let's look at a competitive inhibitor. A competitive inhibitor has a structure that is similar to that of the substrate, right? So if it's lactase, lactose, the inhibitor will look a little bit like lactose, and it will be able to combine. And you can form, when you have lactose, you can form the enzyme substrate complex, and you can get your product of lactose being hydrolyzed. But if you have an enzyme and an inhibitor that looks like lactose but not exactly like lactose, it can bind that enzyme, and now the enzyme cannot bind the substrate because it's binding the inhibitor. It doesn't know it. It just is chemical. It reacts to what its environment is. Now you have, and if you add more inhibitor, there's going to be less and less substrate to bind the enzyme. It's going to probably bind the inhibitor, and the more and more inhibitor you add, the more and more the enzyme is going to bind the inhibitor, because the inhibitor is more available, the substrate is less available, and now you're going to get very little product form, and you're going to block that enzyme. But these things can come off. They compete for the substrate for the active site. They're reversible. As you can see, the arrow goes both ways. So this is not permanent. This is not like this is not so much like a antibiotic such as penicillin. But our body sometimes creates natural inhibitors. The inhibitor could actually be the product. We haven't talked about that. We don't need to at this stage. Competitive inhibition. The enzyme binds the substrate and the enzyme complex is formed, and the product's formed. But if we have a competitive inhibitor, as we just saw, we have the inhibitor binding at the active site, and the enzyme, there's no way to make the product, and this is blocked. We can't make the product, and we may not be able to free up the enzyme too much if there's a ton of inhibitor. So the enzyme is blocked in two ways. It can't free itself from the inhibitor because there's so much inhibitor. There's always another inhibitor molecule there. It can reverse, but you have to get rid of some of the inhibitor to free the enzyme up. I don't expect you to understand all of this, but have some appreciation of this model. What about a non-competitor inhibitor or a non-competitive inhibition? What does that do? Well, it does really the same thing. It stops substrate from being turned into product. So a structure that has a much different than the structure may be way different than the substrate. It's not a pseudo substrate, which is what we looked at earlier. A pseudo substrate. It's almost like the substrate, but it's not, and it can be used to inhibit the enzyme. But now we're going to create a structure that looks totally different, and it binds the enzyme instead of at the active site. It doesn't have to bind at the active site. It can bind somewhere, it can combine somewhere on the enzyme, somewhere else, causing the enzyme to change its shape. And this prevents binding of the original substrate. So let's look at what that might look like. And it cannot be reversed by simply adding more substrate, right? So I can't just add more lactate and overwhelm the inhibitor and just have more substrate molecules and inhibitors. It doesn't work that way because it doesn't bind the active site. It's a non-competitive inhibitor. How does that look? Well, it looks like this. The same process is going on where the enzyme and substrate can combine, and the enzyme releases a product from the reaction of the catalysis. The enzyme does the catalysis when it's in ES mode. When it's in ES, it can catalyze a reaction and form product. But if we have a non-competitive inhibitor, the active site is open. The substrate could bind to it, but when the inhibitor binds at another site of the enzyme, which happens to be here, which is not the active site, it's a site for an inhibitor to bind, it causes the malformation of the enzyme, and the enzyme inhibitor complex is so distorted it can no longer bind the substrate. So it cannot form a product. And there's no way to we could add more substrate, but it doesn't matter. As long as that inhibitor is there, it's non-competitive. The inhibitor doesn't compete with the substrate. So that's the thing where blind competition doesn't always work, right? There's other forms, other things going on, even in politics and in science. Um, Competition is a theory, right? Whether it works perfectly or not is definitely up to debate. And in science, we know for a fact that this is non-competitive inhibition. Wouldn't matter how much substrate I added here for the most part. The inhibitor here is more important. If there's more inhibitor here, it's going to warp this enzyme. It's going to change its shape. And this enzyme no longer can bind this substrate while that inhibitor is attached. And if this inhibitor can come off, that would be good. Maybe there's some way for that. But if the inhibitor is permanent, the enzyme is rendered useless. So non-competitive inhibition can be in different forms. But this is that was a good model. Now we get to my point. Really, I was kind of mixing up um, non-competitive inhibition with irreversible inhibition, and they are a little bit different. And that being that the inhibitors, such as penicillin, as I mentioned, and they show 5 different forms of penicillin. All the structures look like this with the R group, and there's 4 other R groups that can be attached here. Instead of like a hydrogen, you can have penicillin G with a phenylalanine-like character. Here you can have penicillin V attached to this R group, which you add an oxygen or a methoxy-like moiety or an ester-like form. Remember, or not ester, ethers. Remember ethers? That's like an ether. That's not an ester. Ester and ethers are different. Ampicillin, that could be in place of this R group. That has a amine linkage to it, an amine, not amide, amine. You could have amoxicillin, which has an amine group and an alcohol group. All of this can be changed on the penicillin molecule, either made in laboratories synthetically or taken from other bacteria or other sources of defense. Sometimes bacteria can provide uh, in bacteria can sometimes give you um, antibiotic properties. Bacteria has molecules it produces to fight other bacteria off, right? So bacteria may actually contain antibiotics that can be good for humans. Of course, we have the problem with antibiotics being rendered useless and no longer inhibiting certain organisms. So we have to worry about mutations and antibiotics becoming useless because of mutations in bacteria and such. Now, this is irreversible. And when this binds the enzyme, it loses all activity and it forms covalent bonds. Irreversible inhibitors form covalent bonds. Reversible inhibitors form hydrogen bonds and salt linkages that can be broke. Covalent bonds, when they get formed, and now here you know the important reason you learn the difference between a covalent bond and a hydrogen bond. Covalent bonds are way stronger than hydrogen bonds, but hydrogen bonds are very common and do have energy to shape molecules and stuff, especially in their huge numbers. But this is a toxic substance to bacteria, and it can destroy enzymes and destroy bacteria when it's used properly. It forms covalent bonds. This structure forms a covalent bond with the enzyme, making it unable to bind its substrate and cause the bacteria to die. It can also be used in nerve gases and insecticides or antibiotics. We get into dangers of environmental contamination and all these things that need to be thought about as our society progresses. So competitive, non-competitive, and irreversible inhibitors. The characteristic uh, of the shape of the inhibitor, the binding to the enzyme, and the reversibility in competitive, non-competitive, and irreversible forms of inhibition or inhibitors that do such. So summarizing this up, if it's a competitive inhibitor, the shape of the inhibitor is going to be similar to the shape of a substrate. And if it's non-competitive, it may not look nothing like the substrate. And it does not have it if it's irreversible, it may have no similarity to the substrate as well. Um, when we think about competitive inhibition with regards to binding to the enzyme, it competes for and binds at the active site, just like the substrate. If it's non-competitive binding to the enzyme, it doesn't bind at the active site. It binds somewhere else on the enzyme and can change the entire shape of the enzyme, messing up the active site, right? If we have an irreversible situation with regards to binding to the enzyme, it forms a covalent bond with the enzyme and the enzyme may no longer function. Now, what about reversibility with respect to competitive, non-competitive, and irreversible? Well, if in competitive inhibition, if you add more substrate, it will overwhelm the inhibitor and allow the enzymes to start functioning well again. If you have non-competitive inhibition, the reversibility, it's not reversed by adding more substrate, but by a chemical change either induced in the body or artificially to remove that inhibitor. If it's irreversible, the reversibility, of course, is permanent. It's not reversible, and you may have wiped out that enzyme, and the enzyme is rendered useless for all time. So that is a summary of competitive, non-competitive, and irreversible inhibitors. Um, Identify each description as an inhibitor that is competitive or non-competitive. So we're not talking about irreversible, we're just talking about competitive and non-competitive inhibition. So let's see if we could recognize this. Increasing substrate reverses inhibition. Increasing substrate reverses inhibition. Okay, that's competitive, right? So that's competitive. B, it binds to enzyme surface but not to the active site. Well, that's non-competitive inhibition. We learned that changes the active site and it binds away from the active site, so that's non-competitive inhibition. For C, its structure is similar to that of the substrate. Well, we learned for non-competitive, the inhibitor doesn't have to look like anything the substrate does, and it binds somewhere besides the active site. So if the structure has to look similar to that of the actual substrate of the enzyme, that's got to be competitive. Okay. Now, finally, D, inhibition is not reversed by adding more substrate. Well, competitive, we know we can add more substrate and overwhelm the inhibitor and therefore cause the enzyme to function again and make product. But in non-competitive inhibition, the adding more substrate doesn't help because the inhibitor is binding somewhere else besides where the active site is, where the original typical substrate will bind. So since it's binding somewhere else and the inhibitor can look totally different than the substrate, that's got to be non-competitive inhibition. So you have learned a lot. You guys have learned a lot. Let's move on. Our answer's correct. You can double check what I said there to make sure it's right. And oh, finally, we are coming to a close. Let's all get a little bit nostalgic for this class. I love nursing chemistry and nurses, and you guys are, society needs nurses to function properly. It's it's social, it's economic, it's all those things. So I have a great appreciation for what you guys are doing. You're going to be at the front lines and do great things. But protein concept maps, amino acids, proteins, and enzymes. What the heck? Okay, they're all tied together. All enzymes are proteins. All proteins contain amino acids. All amino acids may not be a protein. They may just be a peptide that acts as a drug, as encapsulins. And all proteins may not be enzymes. They may be structural proteins, right? Or transfer proteins, and they're not enzymes. But proteins contain amino acids. They all have a monomer of an amino acid that has an ammonium group that's N plus H3, or a carboxylate, which is got a minus charge. Proteins contain peptide bonds, amino acids. They have primary structures, secondary structures from the hydrogen bonding that make alpha helices and beta pleated sheets. We have tertiary and quaternary structures. Tertiary structures come from R group interactions and disulfide bonds. Quaternary structures come from individual proteins or peptides coming together, individual proteins interacting with each other to form complex of multi subunit proteins, such as hemoglobin, which has 4 proteins associated together to form a complex. These do not have to be enzymes, by the way. You can have enzymes, which can be a single protein or a single peptide that's longer than 50 amino acids, or it can be multiple peptides that make up an enzyme. Um, Proteins, they're used for structure, transport, hormones, storage, and we have enzymes. Enzymes have active sites. At active sites, we form enzyme-substrate complexes to make products, and we have inhibitors that can stop this from happening and slow it down or stop it altogether. There are natural inhibitors. There are exogenous inhibitors that we add. Then we have proteins that can undergo denaturation, as we saw with the eggs, or by adding heat, acids, and bases or organic compounds, we can cause the protein to disassociate with itself. And we can cause changes in the secondary, tertiary, and quaternary structure of proteins. Denaturation usually does not mean breaking the peptide bond, but we have ways to do that that we haven't talked about. This has been phenomenal. I appreciate all of you. Keep in touch and best of luck on the final. Take care. This has been GOBC Chem 114.